This video is a tutorial about a use case workflow from the Playbook Workflow Builder. This workflow was designed to explain MOAs of side effects for approved drugs of the Playbook Workflow Builder. This use case focuses on gaining insights into the potential mechanisms of a drug by analyzing genes that are differentially expressed in response to drug perturbations in human cell lines from the link's L1000 data. It also examines the overlap between these genes and those associated with specific side effects phenotypes, providing a broader understanding of how the drug might impact genes related to the side effects. Mechanism of action (MOA) refers to the specific biochemical interaction through which a drug substance produces its pharmacological effect. In this use case, we analyze the drug side effects and their underlying mechanisms. By default, the disease input is set to atrial fibrillation, and the drug input is set to ibrutinib on the fact that ibrutinib is known to increase the risk of atrial fibrillation. Our goal is to understand if specific genes are both influenced by certain drugs from Link's L1000 experiments that are up or down regulated by drugs and associated with known side effects, based on MGI mammalian phenotype ontology and GWAS catalog. We will see whether the overlap is statistically significant. The result will be visualized using a super Venn diagram. The workflow starts with receiving inputs of phenotype and drug, then perform term searches on Enricher. We will extract MGI mammalian phenotype ontology and GWAS catalog associated with the term search, and convert the phenotype sets to GMT files. The gene set will be assembled from GMTs. For the drug, we will use the L1000 chemical perturbations consensus signatures to get gene sets. Phenotype gene sets and drug gene sets will be concatenated into one table, and it will be visualized in a super Venn diagram. The default settings include atrial fibrillation as the disease input and ibrutinib as the drug. You can enter any phenotype and drug of your interest. You can utilize this web page to discover potential drug-disease interactions for Playbook use case 2. Starting with the input disease, atrial fibrillation, Playbook Workflow extracts gene sets that contain this disease in the set label using Enricher. We will extract phenotypes from two databases, MGI mammalian phenotype ontology and GWAS catalog associated with the term search. Playbook Workflow converts Enricher phenotype sets from both databases into GMT files and uses them to compute consensus gene sets for MGI mammalian phenotype ontology and GWAS catalog. With gene sets from MGI mammalian phenotype ontology and GWAS catalog, the Playbook Workflow assembles a GMT file. From the input drug, ibrutinib, Workflow extracts gene sets containing the ibrutinib in the set label using Enricher. We extract L1000 chemical perturbations consensus signatures from the term search of Enricher. The Playbook Workflow converts the list of drug sets to GMT format, including a description and gene sets for each drug set. After assembling the GMT files for the drug and the two phenotypes, we combine them into a single file. After merging the phenotype and drug gene sets into a single gene set library in GMT format, we can create a super Venn diagram. A super Venn diagram helps us visualize shared genes across different gene sets. The super Venn diagram is interactive. You can select specific areas to view genes and terms. Click on the areas of interest in the diagram, and a list of genes will appear on the top right. You can also check selected terms in the bottom left corner and easily copy them to your clipboard by clicking on the icon. As a result of the workflow, we have identified common genes that are either upregulated or downregulated by the drug ibrutinib, and these genes are also associated with MGI mammalian phenotype ontology and GWAS catalog linked to atrial fibrillation. This discovery holds the potential to enhance our understanding of the underlying mechanisms connecting the drug ibrutinib and the condition of atrial fibrillation. With that, I'd like to acknowledge the members of the Playbook Partnership team from xRNA, Icahn, Kids First, Links, and Metabolomics Workbench for all of their work over the past year. I'd also like to thank the CFDE community and our NIH program officers for their continued support. Thank you all for your attention.